So Tony is a Cambridge man. He was educated in mathematics and computing here in Cambridge. He then did a PhD at the LMB in crystallography. And then he joined Aaron's group to work on image analysis of electron micrographs. And he focused mostly on icosahedral viruses and also on tau filaments, as I mentioned earlier. From 1974 to 2007, he was a group leader at the LMB. And from 1994 to 2005, he was the joint head of the Structural Studies Division. And in fact, he was involved in hiring me at the time.
We get some things right.
Let's see about that. And he's an EMBO fellow, and he's a fellow of the Royal Society, and he's a recipient of the Nobel Prize of the Royal Society.
Yeah. This system is completely different than the one I have in the lab and the one I have at home. And we're there.
Use that if you can.
Oh, okay. All right. Well, good morning, everybody. This is going to work. In 2001, it was suggested that John Finch and I might conduct an interview with Aaron as a record for the LMB archives. John and I drew up a list of topics that we thought would be interesting to discuss. And on a cold December day, we all went to the university recording studio, expecting to spend a couple of hours talking. After 4 hours, we were about halfway through our list of topics. So we came back the next day and shot another 3 hours of tape. So the tapes have never been viewed in public. So I thought that this meeting might be an appropriate place to air them. Obviously, I can't show 7 hours of video. You'd all get bored stiff. But I've extracted a few short pieces trying to cover a range of Aaron's interests, the sort that we've already heard of from Jan. There will be a little duplication in some of the things that I say. And because they're rather necessarily disconnected, I'm hopping through his career. I've called them snatches of a conversation from 2001, because that's how I believe they come across. The clips are taken from a running edited tape, so I'm afraid they contain timing information which we can't remove. But I'm grateful to Neil Grant, who's here taking photographs, for extracting these clips. So, as we've already heard, I'll try a little introduction to each of the clips. Aaron, as we've heard, was born in Lithuania in 1926, moved to South Africa at the age of 2, graduated from the University of Witwatersrand, taking maths, physics, and chemistry. And then wanting to extend his knowledge of physics, he moved to the University of Cape Town for an MSc with R.W. James, which is a name that will come up a number of times in these clips, who was at that stage a well-known X-ray crystallographer. He then decided to do a PhD in Cambridge, hoping to move into a bit more biological sort of subjects, as Jan has already mentioned. And this is described in the first clip. So what prompted the choice of Cambridge eventually? Why did
well, I thought of going, so Coulson offered me a place at King's College to go into quantum chemistry. Later he moved to Oxford Institute of Mathematics. But I went to Cambridge because I thought I would then go to work in the MRC unit. You see, James was in contact. He kept up his correspondence with Bragg. I should say, returning to James, when James came back from the, from the Antarctic, the war was on, and he went and joined a sound ranging unit which Lawrence Bragg had set up. So they became colleagues. And later he went to Manchester with Bragg and did those famous experiments on Bragg, James, and Bose-Kay on measuring zero-point motion and things of that sort by X-ray crystallography. So Bragg and James were friends. Yeah. And then he'd been receiving letters, and James told me that he'd had a letter from Bragg about the progress they were making with proteins, and there was a man called Kendrew, he mentioned, who was now crystallized myoglobin, which was a small versions, I think he put it, of hemoglobin. So I decided that I would go to Cambridge and brag. So James, I got a 1851 exhibition scholarship. I also got a research studentship from Trinity, I think on the strength of my molecular structure work. I should say I didn't give me both. They took away. It was only, I didn't have two fellowships.
You probably had rooms, did you?
No, I was a research student, so there was no rooms. Research students hardly existed. Undergraduates, as you well know, were the prime source of, were the prime objects of teaching in Cambridge. So Trinity didn't have any rooms for a research student, particularly a married research student, because then we got married when we went quite young, you see.
Yes.
And my wife, Liba, was going off to be a dance student in London. Well, originally it was going to be Cambridge, because the US Lader School of Modern Dance was actually in Cambridge during the war and some years later, so we thought we'd both
Was that an
go
additional
there.
reason for choosing Cambridge?
Yes, I think it was. But I think I would have chosen Cambridge anyway. So I applied to join the, so basically James sent me to Bragg. That's really, if you summarize it. And so I went to see Bragg, and Bragg told me that the MRC unit was full, so I couldn't work on proteins. But I did want to work on some unorthodox problem of structure of matter. And I'd read quite a bit about order disorder in alloys and malloys, and I found it quite interesting how you get segregation in alloys. But how does it happen? You know, when you change the temperature and the segregation of different phases. So he said they'd stopped that kind of work. There's a man called Hargreaves that published.
So that's the first clip of Aaron explaining how he came to be in Cambridge. After his PhD, still wanting to move into biology, he got a fellowship, as we'd heard, to work with the protein crystallographer Harry Carlisle at Birkbeck College in London. And that didn't work out very well, so Aaron started to collaborate with Rosalind Franklin in the same department. And this led to Aaron's first major biological interest, the structure and assembly of viruses. And as you've already heard from Jan, in 1962, he and Don Casper published their classic paper on the geometrical structure of icosahedral viruses. As a side note to this paper is actually Aaron's most highly cited paper. So it was based on the idea of taking a hexagonal sheet. It's not working. Oh, no. I can't see any light from that. Anyway, taking a hexagonal sheet, cutting it to introduce some fivefold axes and folding it up in a symmetrical way. And this could build icosahedral shells of arbitrary size. And the next clip.
How do you build a virus? Well, you try to use as many subunits as possible, and therefore you have the highest possible symmetry. Now, in the plane, the highest possible symmetry is a six-fold symmetry, six-fold axis of symmetry, a three-fold axis of symmetry, and two-fold axis of symmetry, which a crystallographer would call 6-3-2. So if you now change one of the units, one of the six-fold units, into a five-fold unit, you can have the local geometry is much the same, but you now have a 5 instead of a 6. When you do that, you introduce curvature. So therefore, we argued that the virus structure shell should have five-fold, three-fold, and two-fold symmetry. And that led to the prediction of the different structures you could have.
All the T-nums.
All the, what we call T-num, doing a systematic, by choosing your 5. You had to have 12 fives and only 12 fives in order to close the shell, the curvature. It all comes from a simple mathematical theorem called Euler's theorem. And you could have as many 6s as you liked. not as many, but there would be a fixed number of 6s. And so we produced
really
it.
the first general picture of how virus
Yes.
structure with more than 60 subunits
Yes,
worked.
that's right, yes.
So after that, in 1962, again, as Jan has said, Aaron moved from Birkbeck to the newly opened LMB here in Cambridge, and his main interest remained in the structure of viruses. And the way of approaching this at that stage was through the use of negatively stained electron microscope samples. And this shows some of John's pictures showing the kind of reproducible features on the left that they were identifying that they could see, the micrographs in the middle column, and then some later computer simulations suggesting how these patterns arose. And the interpretation of the images of negatively stained specimens were very much open to question at this stage. Were they plaster casts or footprints? And from John's images of tilted specimens, it became clear that the particles were embedded in stain, and the image were therefore represented projections or superpositions of the structure. So this is then Aaron. You mentioned electron microscopy in connection with Hugh. I mean, what are your memories of the early days of electron microscopy?
Well,
it came
people
to play
had
a very
really
important role.
Yes, it did, and I realized from the spectacular pictures obtained by Horn and Brenner on adenovirus, which was called negatively stained, that would be a powerful tool. And just looking at the outline of the virus structure, and there were quite a number of papers on the subject, people didn't understand the relationship between the knobs, or the protuberances they saw in electron micrographs, and the underlying structure. And I remember having to define chemical units, structural units, and morphological units, which was an important distinction at the time, because people were very confused. So chemical unit was the basic chemistry, a structural unit could be a cluster of chemical units, and morphological unit could be a further grouping of structural units. And in
Basically what you saw in the microscope.
Exactly. And so, for example, turnip-yellow mosaic, as I mentioned earlier, had been shown to be made of 32 subunits, but the relationship to the chemistry wasn't at all clear. And I told you. And I remember looking at the turnip-yellow mosaic virus pictures, and they were all so different. Now, what we thought was that the particles were being distorted, and what we have been thinking of, and this is what Hugh Huxley had been thinking of, and also Horn and Brenner, that when you did negative staining, the virus particle was making a footprint in a bed of stain. And I think we came to realize that this couldn't be happening at all. What was happening, first of all, on the chemical grounds, since the stain is hydrophilic, it would surely cover the virus surface. I came to realize that this it wasn't a footprint, but a cast, and therefore you were looking through top and bottom of the surface.
When the idea of a superposition
yes,
pattern
a superposition pattern, yes.
I remember those.
And we drew drawings and began to realize what they were, that they were superpositions. And then John did some classic experiments in which he tilted particles and the view changed. Well, that meant, and then we could learn to interpret them in terms of superpositions of the top and bottom surfaces. And that made, that was the beginning, and so we were able to interpret quite a number of virus electron micrographs. And of course we had a theory of virus structure, so for example there were quite a lot of papers published on perleoma virus, and they were all wrong, and I remember writing a paper pointing out what the real structure was. And the only time I ever had a compliment from Sidney Brenner in all those years was when he said, when we published our polyoma, interpretation of polyoma, he said, see, the words to the guys, oh, you slaughtered those guys.
I think probably Sidney said other things to Aaron over the years as well. Anyway, so having established that the images represented superpositions, it then became a question of whether it was possible from the images to separate the different layers in the structure. And by creating an optical diffraction pattern, the far image is an electron micrograph of a polyhead, which is an aberrant head from T4 bacteriophage. If you make an optical diffraction pattern, you get two sets of spots that can be imaged corresponding to the two different layers of the collapsed tube. And by letting one or other of the sets of spots through, you can create a filtered image of what they call the near side or the far side. And you can see the hexagonal packing of the molecules in the tube. So the next clip deals with this kind of filtering.
And it was this sort of stage that the idea then of manipulating the image by filtering it came.
Yes, that's right, yes. Because I realized that if you could see back and front separately, you could show the, you could sort out an image like this. And this is a tubular structure, which is a mutated version of the head of a bacteriophage with arrays of subunits. And you could interpret this in terms of the overlap of the outer surface and the back surface. And you could sort out the diffracted rays which belong to one surface or the other, and showing one side of a surface. Now this met with great suspicion. Kellenberger, who was the distinguished microscopist, said it was all artifact. I remember I went around saying it because we had created this image. I did say we had created out of the beams that were present, and we showed the two-sided images. So this was the beginning of filtering, optical filtering. And we did a number of trial experiments as well with TMB and things of that sort. So that was manipulating an image optically.
Then how did the idea of that then develop into sort of a full three-dimensional reconstruction?
Well, that was related to the tilting experiments we had done. You see, when John Finch was tilting the spherical virus particles and showing the images changed, we were building images predicting what the things would look like in a computer. So clearly there was enough information in the pictures when you tilted them to define an object, or the structure of an object. And so that was happening at the same time as the optical diffraction of these linear cylindrical structures. The two things were hand in hand in the same lab. And when we were doing the structure of the bacteriophage tail, which we were looking at this is the tail of the bacteriophage through which the DNA of the bacteriophage is injected into the host bacterium and infects the host bacterium. It's a bacterial virus. So I don't remember why we chose this object particularly, but this is the one in which I began doing simple filtering and treating it, although there's not these spots were treated in a way as though they were two-dimensional, saying that most of the contrast would come from the front and the back surface. So we began filtering, and this is a filtered image, optically filtered in that way. And I could see on this two systems of stripes, which are the fronts of a helix. And yet on the inside we could see another system which had a different slope. And that was the key. I then realized that these must be two systems of helices on the let's say on the front surface, but one on the outside of the particle and one on the inside, more closer to the center, and they would have different slopes. So I realized we were actually looking at a three-dimensional image, and that this image contained the information for three-dimensional structure. That was a key point.
Well, the question then became how they could actually compute a three-dimensional map from the micrographs. And you can see top left there are the two-dimensionally filtered images with the sloping helices of one side showing the inside and the outside, and then a three-dimensional map here. These are taken from the classic paper of David Rosier and Aaron in 1968. The methodology, the mathematical basis, was based on the projection theorem, which has been mentioned already, which says that different projected images seen in the microscope, when Fourier transformed, give you central sections of the three-dimensional transform, which can then be inverted to make a three-dimensional map of this kind. And Aaron goes into this a little bit more in the next clip.
Well, I was never quite clear, even because I wasn't involved directly at that stage. Where did the notion come from that by computationally Fourier transforming a digitized image, you recovered the phases? Whose idea was that? I mean, it's sort of obvious looking back, but...
No, we see, it puzzled Max. Max kept asking us, Perutz, where do the phases come from? I said, the phases are in the picture. Because the diffracted beams, we knew that the diffracted beams here carried amplitude and phase. That was very clear to us.
But how was the connection made to computing that numerically rather than...
I think David took the first step to say that...
David's idea, because he got so fed up with measuring phases optically.
No, I knew you could do it, but I thought that would be quite a... Because Fourier transform was very, very slow. We had used beaves, ellipses, and strips. And I thought that would take forever. And I remember saying that this wouldn't be any short, it would be more elegant and well as practical. And so the idea of measuring and calculating a complex Fourier transform with phase, that was but this was, in the first instance, we did this for helical structures where we commanded the density. And then I thought, well, this is all right for a cylindrical structure. What about a spherical virus? So I tried to do the same thing. I tried to expanded the density in terms of spherical harmonics, in terms of icosahedral spherical harmonics indeed, and then looked at the projections. Now the projection of a spherical harmonic can be worked out, but it's fearfully complicated. And I thought this can't be. This must be part of a more general. And David was also puzzling about this, and I realized that the application of something I had known for many years, because I had solved X-ray structures of organic crystals using what's called the projection theorem, where a slice of the diffraction pattern, a slice in Fourier space, as we would say, or reciprocal space, as Klug would call it, represents the projection of everything down the axis. So to the projection theorem, which you could write down a couple of lines of mathematics.
So was that a penny that suddenly dropped, and you sort of in your bath and you thought,
penny
my
dropped.
God, the projection theorem?
It was. It's exactly that. Exactly that.
Sure where that froze, but I think we got to the end of it. Anyway, I've dwelt at some length on these early developments in interpreting electron micrographs, because I think, as Jan said, this was one of Aaron's most significant contributions. And David de Rosier, in the next talk, I think is going to say quite a lot more about the topic and some of the things that I've just covered briefly, and that Aaron has also covered briefly. So Aaron later worked on a number of different biological systems, including TMV structure and assembly, transfer RNA, as we've heard, and we'll hear more about that later, and tau filaments in Alzheimer's disease, which again we'll hear more about later. So I've just chosen a couple more topics to illustrate his range of interests, namely nucleosomes and then zinc fingers. So I'll go fairly quickly through this. Nucleosomes, as you all know, are the packing units of DNA and chromosomes, with DNA wrapped around a core of histones. These are some of the early structural results on nucleosomes. On the left, there's a map of the histone core computed from micrographs of tubular assemblies of histone octamers. And on the right, there's an X-ray crystallographic map at 7 angstroms resolution, showing DNA brown wrapped around the histone core in blue and green. And in the next clip, Aaron talks about the early work on nucleosomes.
I think they still thought of it. They had no idea that this would be, thought of it in terms of beads on strings. But you see, chromatin is not a bead on a string, it's a string on beads. Although I try to propagate those words, it's never taken off. But really is a string on beads, of histone octamers. And anyway, Roger published his paper with Gene Thomas in Science in the end of 1974.
And this established the idea of a nucleosome.
A nucleosome, but that name was given later by
Yes.
Chambon, who defined the nucleosome as that which entity which contained a repeating unit of DNA, which was a great nuisance because it's typical French. He wanted a definition at an early stage before we knew anything. See, Francis
you were
and I,
defining.
we called them beads. But he said you have to have a precise definition, otherwise you don't know what you're doing. You know, typical, like Lvov had done with capsomeres and years before. It's all the Cartesian French. And so, unfortunately, it turned out the repeat of DNA varied among different tissues. So there was clearly some issue. What was the real repeat unit, or the real structural unit? And so this emerged from some studies when we decided to make an effort now on crystallizing the nucleosome core particle. I'll explain what that is. That's the particle. If you digest chromatin for micrococcal nuclease, it cuts the DNA between the particles, and eventually it trims it down to a small particle called the core particle. It's done in stages, and this was the work of several people, but Len Latta, who was a postdoc in our lab, showed you could get a population of pretty homogeneous population of about 146 base pairs associated with a histone octamer. And the fifth histone, H1, which was rather mysterious, nobody knew quite where it was and what it did, was missing, or had fallen off. And you could see this on gels. And then a critical experiment, there were two critical experiments, well, one critical experiment of the nucleosome, where John Finch, Marcus Nolan, and Roger Kornberg, in the tradition we'd established for TMV, decided to find out what was the structural unit. And they chopped the chromatin down in a series of stages and ran the results on sucrose gels. And they were clearly multiples. There was a unitary size, one which was double, one which was 3 times, one which was 4 times. And in the electron microscope, John showed, I should say, the fact that we got crystals with mixed DNA sequences or random DNA sequences did surprise all the molecular biologists, because they thought, they couldn't conceive that there was something highly ordered as nucleosome particles inside the nucleus. But they always, psychologically, they thought of it as some kind of spaghetti DNA and so on. And so this was important, I think, in the field to show that there was order even at this level in nature. And there was a series of levels, because by now we'd worked out the first level, the nucleosome core, then H1 comes along and ties the two ends of the DNA, which have been encircling the nucleosome, and mediates the formation of the next level, the solenoid structure.
Shall I do the zinc fingers one, or shall I skip the next one? Is that okay? Okay. So transcription factor 3A occurs in large amounts in oocytes, where it controls the transcription of ribosomal 5S RNA. And it binds to both the 5S RNA gene and the 5S RNA itself. And that dual binding property suggested to Eric be interesting as a structural problem. The protein extracted from Xenopus oocytes contained a large amount of zinc, and on digest appeared to have a repetitive structure. And when the protein sequence was published, it proved to have repeats containing pairs of cysteine and histidine, which coordinate the zinc. And in the next clip, Aaron refers to this diagram on the left, bottom left, as looking like frogs. It's this little joke.
So about this time, the protein sequence came out.
Yes, but only after we had shown that the protein contained about between 9 and 11 zincs. We did absorption spectrophotometry and absorption spectrophotometry and later XFs. So the sequence came out, and so we knew that, and we also had done biochemistry. It wasn't just the zinc content, that in the standard way, when you got a new protein, you try to chop it up and see if there was a domain, some domains, as many proteins are made of domains. and Jonathan began using various trypsin, chymotrypsin, and he found that he could get down to roughly a 3K limit digest, and the molecule is 40K. So it looked as though there was something which was resisting degradation beyond 3K. And I asked him if there was any sign of multiples of 3K. He said no. But he was very messy, so I looked through, he didn't through his things, I looked through and I could see with my own eyes that there were real bands. And he'd been told that by chemists you always get these intermediate bands. So I got him to plot them out, and they were on a periodic. And so I realized that we had multiply repeated units, and of a size of 3K. So it was very straightforward. So when the sequence came out, I looked for, in the pattern, I looked, and by this time we'd shown there was a metal, and I looked for, by hand, for repeats, and I could see there was some kind of repetitive sequence of cysteines and histidines. And I couldn't get it all out, but I still have the piece of paper where I traced it. So I rushed over to Andrew McLaughlin, who had these sophisticated programs, and for 1 hour he produced an analysis and showed that there were 9 repeating units, each of which contained 2 cysteines and 2 histidines, which are the natural ligands for zinc.
You call them zinc fingers in the
we called them, no, we didn't. But we called them, the word fingers came about because we realized they were gripping or grasping the DNA, not because they looked like a... In fact, the drawing, you'll see it looks like it's a frog, a xenopus. It was done deliberately, actually, a bit of a minor joke. And
why not?
So just one very long last clip, which is quite short.
Are there any people that you feel particularly influenced your career, early on? People you...
Well, I think R.W. James in particular. R.W. James, where I learnt what, it was I think the British empirical tradition in Cape Town, Cambridge tradition in particular, Manchester I should say, Manchester and Cambridge. And the other person who influenced me, I think, was Rosalind Franklin, from whom I learned the virtues of single-mindedness, although I didn't conscious at the time. And I think, but above all, Francis Crick, not by what he had done, but by watching the way he went about a problem. And even though he never published anything on chromatin to speak of, the way he approached it, and even his error, his wrong paper in Nature,
was fertile somehow.
Fertile, yes, and I've quoted from Whitehead that it's more important that an idea be fruitful than it be correct. And in my Nobel lecture, I put in a picture of the original hypothesis of how TMV assembled. The editor wanted to cut it out. He says, you can't put that in, it's wrong. I said, but I say it's wrong. And there that was a good starting point. And he agreed that. So I've learned, but I think of all those individuals, those 3 have influenced me scientifically. But I don't think I had any heroes, really. I've also seen a number of people in the world, scientists, learned what not to do, but they will be nameless.
They better be.
So that's all the clips. I'm afraid I've run over slightly, but I hope they've served as a reminder of Aaron for those who knew him, and as an introduction for those who didn't. And a lot of the topics that have been touched on here will be covered in greater detail by the later speakers. And I'll leave you here with Aaron in relaxed mode in his garden. Thank you very much.
Thank you, Tony. Very exciting to hear Aaron talk about the moment when they realized that electron micrographs have 3-dimensional information.
Indeed.
Since we are behind time already,
Sorry.
that's all right. Maybe we can have one question if there's one. Thank you, Tony.